The next speaker, who is Alice Ruixue from the University of Oslo in Norway. Hello, Alice, can you hear us?
Hi, everyone.
Whenever you're ready, you can just start with your talk.
Hello, everyone. Thank you for the host of Aging Research and Drug Discovery meeting, giving me this opportunity to have a short talk here. I'm Alice Ruixue Ai from the Fang Lab. I will give a talk on defective mitophagy in Alzheimer's disease and AI-based drug development. There are three main topics that we are working in the Fang Lab. The first one is working on a small molecule called nicotinamide adenine dinucleotide NAD+, looking at its roles in health and longevity. Second, we are also interested in Alzheimer's disease, since it's very common and age pre-post. We are looking at how AD is affected by impaired garbage clearance capacity called autophagy, especially in the mitochondria, where it's termed mitophagy. Finally, we are also interested in translational medicine. So we are working with colleagues to develop AI-based biomarkers to develop drugs to tackle AD and aging. We are interested in AD is because Alzheimer's disease is a formidable healthcare and socio-economic challenge. It's the most common form of dementia. Around 50 million people are suffering from this disease worldwide, and the number may even grow to 92 million in 2030. It's estimated that it will cost society $2 trillion by 2030. The name of disease has been defined for over 100 years. However, there is still no cure. The reason is that AD is really complicated. In the left corner here, we can see a normal brain. However, compared with the AD brain, you can see cortical atrophy, enlargement of lateral ventricle, and the shrinkage of the hippocampus, as well as the anterior cortex, which are very important for memory. There are two disease-defined pathological features of AD, A-beta and tau tangles. So based on these two disease pathologies, people have developed a lot of drugs to treat AD. However, all of them have failed so far. So what we have now is a situation where there are increasing numbers of people are suffered from this disease. However, no effective treatment to help them. This is, of course, a big problem. So we wonder whether we could use the knowledge gained from aging research to help us understanding AD. It is because aging is indeed the greatest risk factor for AD. Recently, a study came out from Fang Lab, the NIA. They did a careful evaluation of brain anatomy from both healthy people and AD patients. And in the top image, you can see a slide taken from the hippocampal region of healthy people. We can see this intracellular structure. We call it mitochondria here. It's produced the energy needed for human to survive. Here we can see a healthy mitochondria with a complete membrane. Together, when we check the brain of AD patients, we show a great hole here in the mitochondria. They tangled together, possible due to the effective removal of the garbage elimination system called mitophagy. The concept we have been working with is that when we are young, we have an effective mitophagic to quickly clean the garbage, making our brain and body healthy, sort of like the engine in a nice and new car. However, when we are getting old, the machinery starts to become damaged and gets less efficient, leading to an accumulation of garbage, which is bad for the brain. So for the last 5 years, Evandro and his colleagues have been studying this phenomenon using a cross-species platform, including human brain, iPSC, ARDD mice, and C. elegans. The hypothesis is there are different factors that can contribute to impaired mitophagy, resulting in an accumulation of damaged mitochondria. At the same time, other risk factors like tau and A beta, ApoE4 also help play an important role in the process, forming a very complicated network. However, all these factors can contribute to neural dysfunction and dysfunction of mitochondria. Finally, they may contribute to brain atrophy and memory loss. Here we show the mechanism that could contact to impaired mitophagy. This is important for AD because we found upregulation of mitophagy can result in an improvement in mitochondrial quality, which can then increase neural function through important survival and healthy microglia. Mitophagy upregulation can further contribute to reducing tau and A beta, which means may possible to improve the brain health. In 2019, Evandro and his colleague published a paper in Nature Neuroscience indicating mitophagy may become a new powerful candidate for treatment. With this in our mind, we developed the AI-based identification of potent mitophagy inducer as an anti-AD drug candidate. We use the scale of artificial intelligence and using AI to screen drugs for AD research. And the project was led by Dr. Chenglong Xie. I was very lucky to be involved in this project. We collaborate with the colleague from the University of Macau, Imperial College London, and MindRank AI. Actually, AI has been used widely in healthcare and related fields to help sort large amounts of data. Here we use the AI to screen a large number of small molecules that can induce mitophagy, then test their treatment effects on an AD model. Today, I will give a summary of what we did. Here is a schematic abstract of this project. Firstly, we target mitophagy inducer. Then we use the AI to examine the chemical properties and the formation of a large number of non-mitophagy inducers. After identifying a number of potent candidates molecules, we use the wet lab, including cells, C. elegans, mice, to validate AI-positive drug candidates. We were very lucky to get 2 lead compounds. Here we labeled K and R. Speaking of the AI part, we used the unsupervised machine learning model, including vector representation of molecular structure, pharmacophore fingerprinting, and conformer fingerprinting. We first got access to the drug library from the University of of Macau. After that, we examined the molecules in various dimensions using molecular representations and the retrieved models here. We then look at the results as an aggregation using hyperspace filter. The results of this process was the identification of several new molecules for further research. In summary, by exercising this molecular library, which contain around 3,274 molecules using a cutoff value of 0.75, we were able to choose the top 18 compounds. Then we tested the compounds with Helakema cell for mitophagy, and we found totally 7 compounds were able to induce mitophagy. So after that, we asked whether these 7 compounds could induce mitophagy in neurons as well as the cells. We use a simple animal model, C. elegans, to test their function in neural regeneration and memory improvement. After this phase of testing, we had 2 inducers. Finally, we tested these 2 inducers in AD mice. We found that both of them can improve memory and reduce tau and A beta. Here I want to show some of our data. The left is the water maze results. Water maze is a way to evaluate spatial memory and learning in mice. It tests the timing. It tests the time it takes to a mouse to find a little platform just underneath the surface of the water. After treating the mice with K and R, the time it took to locate the platform decreased, indicating they were learning better. On the right here, we can see the p-tau results. You can see the expression of various kinds of p-tau decrease dramatically with the treatment of K and R. To answer specifically how these inducers could be working, we look at their effects in neural line with the treatment of K and R. We can see the reduction of p-tau 181 compared with the control group. However, when we knock down mitophagy gene, pink one here, it dramatically impeded the p-tau 181 inhibition capacity. And the p-tau 217 shows the same pattern. The data shows that the AI approach is effective and able to specifically target ways to improve AD pathology, while at least partially mitophagy pathway. So to summarize, machine learning enables fast and accurate identification of potent mitophagy inducer. K and R stimulate neuronal mitophagy, improving memory in A beta and tau, expressing in AD models, including C. elegans, AD mice, in a pink one parking dependent manner. Mitophagy in decrease tau pathology through inhibition of multiple p-tau sites, including p-tau 181 and p-tau 217. Defective mitophagy is a risk factor for AD, and mitophagy may be a drug target. Finally, I would like to thank all the members in the Fang Lab, all the collaborators. Thanks for your time. And I'm very happy to take any questions.
Thank you very much, Alice. Maybe we have time for a very quick question. There's one from James Payer. Do we know which cell type mitochondrial function is important for in the AD context? Neurons, astrocytes, microglia, endothelial cells?
all the cell lines are very important for texting a NAD+.
Okay, I would recommend you just to go to Slack. There are several more questions. You can answer them there. And thank you very much for joining us today.